application of crossover frequencies, which is being able to build chromosomal maps. And our criteria is being able to actually make a chromosomal map out of crossover frequency. So you'll remember that I mentioned that the frequency of crossover depends on the distance between the chromosomes. Now, we don't actually want to measure that on a nucleotide by nucleotide basis because that is tedious and that is incomprehensible because of the number of nucleotides there are in a given chromosome. However, scientists can do something different. We use a completely different unit, which is map units. And what each map unit is in a chromosomal map is one map unit represents a 1% frequency of crossover between linked alleles. So if we know that, like our last video's example, there's a 17% crossover frequency, that would mean we could predict it's about 17 map units apart from each other. And exactly how far a map unit is will depend on the species and the frequency of crossover within that species. Scientists can use this fairly effectively. This is a map of one fruit fly chromosome with a whole bunch of different alleles mapped out on it and genes mapped out on it. And you can actually see there's different locations for eye color, for leg length, for body. And we see how many map units there are on these. And note, this diagram is not to scale. You can see that because you have long wings at 13 map units, and then long legs at 31, and then gray body at 48.5, and it's totally not to scale. But that's okay. We don't need it to be for our purposes. So, from this map, we could predict the crossover frequency between the long wing dumpy wing gene and the long leg short leg gene is about 18% because we're going 31 minus 13, which are the two locations. You'll note that all of these numbers are based off of the long feeler short feeler gene right up at the top. Now you'll also notice something really interesting about this map. At one end we have the long feeler short feeler at 0, and at the other end we have red eye brown eye at 104.5%. You can't have 104.5%. It's not possible. You can max out at 100%. So what does this mean? Well, that means that that's how many map units there are apart from each other. Basically what it's saying is because these two genes are on opposite ends of the chromosomes, the chances of them crossing over are so high that it's practically a guarantee. Now, why can't we just say 100% for that? Well, the 104.5% keeps it relevant to other nearby traits. So yes, you can get genes that are more than 100 map units apart from each other, which means sometimes we can translate those map units and, oh, this is likely to be greater than 100% chance of crossover because they're so far apart. So for example, between the red eyes and the straight wings, that one is a measurable distance. That one you would have 29% difference between the two of them. So 29 map units apart. So even though the red eye brown eye distance is kind of weird when you compare it to stuff at the other end of the scale, as long as you keep comparing genes that are close to each other, everything works out. If you start comparing them really far away, some weird stuff can happen. And really, past 50 map units apart from each other, you can't tell if it's actually on the same chromosome. And the reason for that is because independent assortment, you have a 50-50 chance of the genes going together or going apart. So if you see there's, oh, this is 50-50 or this is greater than 50-50, what that likely means is that, well, you don't know. It could be on the same chromosome, it could be on different chromosomes. So the red eye, purple eye gene and the long feeler, short feeler gene, you can't compare those two and see, are they on the same chromosome? But what you can do is you can link them all together. You can compare it to other genes that both of them are linked to. So like, look at the long legs, short legs. You're going to have about a 23.5% between long legs and red eyes. And of course, you have a 31% between long legs and long feelers. So that would mean that those two are connected to each other. Even though you can't see that red eyes is connected to long feelers in most cases, because the crossover rate's too high, you can see with the lower crossover rates, hey, both of these are connected to long legs, and then the long legs together ties everything together. So basically what this means is that the crossover frequencies in between add up to create the distance between them. Now, in reality, we're measuring it from the end of one gene to the other one. So you see in this one, we have 3 genes, B, CN, and VG. The crossover frequency between B and CN is 9%. Crossover frequency between CN and VG is 9.5%, but then the crossover frequency between B and VG is 17%, which is less than the two of them added together. Well, that means that the CN gene is probably 1.5 map units long. Now, on diploma exams, on tests in high school, don't worry about this. Assume the length of the gene is not going to influence the overall result of crossover frequency. But yeah, it's likely that what's happening here is that there is a little bit of a gap in between the added up numbers because of the sizes of the genes themselves. And of course, we can always just figure out the frequency of crossover by figuring out how many recombinants there are and dividing that by the total number of offspring produced in a given generation.